This study essentially is also based on one of the high-risk group, which is 1q21 amplification patients. So because within this chromosome, there actually, when the region that is being amplified encompasses a large chunk of genes. And we would like to elucidate what kind of genes that are actually driving the poor prognosis behind the patients. So in this case, we went on to look at both genes that are not being so well studied but are biologically relevant to the disease pathogenesis, which namely the IL6R gene as well as the ADAR1-P150 gene. So we actually identified both of these genes have an regulatory feedback loop that actually regulates the IL6 signaling in order to amplify the oncogenicity of this signaling pathway that could actually confer poorer prognosis in the patients. So essentially the methods that we used was, firstly we did this clinical analysis by analyzing a large or rather a few publicly available myeloma data sets that actually reveal the close relationship between the OA expression of both of these genes, namely the IL6R as well as ADAR1-P150, that actually was also associated to the poor prognosis in this high-risk group of patients, which is the 1q21 amplification patients. So, right. And for the biological part of it, we essentially utilize a lot of molecular biology techniques, which includes the OA expression as well as the knockdown studies by using lentivirus manipulation, as well as by overexpressing and knocking down this both of these genes individually. We actually saw that this IL6 pathway signaling, IL6-STAT3 pathway signaling was largely affected, and essentially it kind of like regulate the cellular phenotype, which show that the differential expression of both of these genes could actually confer a more proliferative nature of myeloma cells. 1q21 amplification patients, they are of high-risk disease. So in this case, we identified that both of these genes could actually confer poor prognosis in the patients, indicating that both of these genes could be therapeutically targeted in order for us to have a more precision, precision medicine is the way to go for now. So the identification of this, the critical role of both of these genes in the myeloma patients of harboring 1q21 amplification clearly indicates that we could actually target both of these genes in order to therapeutically better target this kind of patients, for the high-risk disease.